Well, again, welcome everyone. My name is Jerry Bowers, and it is my pleasure to be with you this evening to welcome an old friend of the MDI Biolab back to our Science Cafe stage, Nishad Jayasundara, whom I think probably a number of you know. And we are very excited to welcome him and have him join us this evening to update us on some exciting work that he is doing. So I'm going to turn things over to my colleague, Fred Bever, who is going to make the introduction and who will be our host for the evening.
Hello, everyone. Thanks, Jerry. And thank you, Nishad, for being here, of course, and for everyone who's viewing now. As many of you know, Nishad has been connecting the dots between human-driven climate change and human health, and more recently seems to be focusing on children, and that's why we named this Kids Climate Change and Kids Health. His most recent appointment is as the Julie Plant Granger Assistant Professor of Global Environmental Health at Duke University's Nicholas School of the Environment. How long have you been in that position, Nishad?
3 years now.
For 3 years?
Yeah, a little 3 years.
Before that, he was assistant professor at the University of Maine School of Marine Sciences. He earned his undergraduate degree at College of the Atlantic. I think we might have some COA people joining here today, which is great. He got his Ph.D. at Stanford. He's been a research assistant at the lab and a visiting scientist, a collaborator, particularly with Dr. Ian Drummond, who is at Patricia Hand's place today. And that really is the thumbnail sketch of a scientist at work. And so now we can hear some of the in-depth stuff. Thanks very much, Nishad.
Oh, thank you. Hope everybody can hear me and see the slides well. So thank you again, Fred and Emily and Jerry, in organizing all of these. It's always a pleasure and an honor to be invited back to MDIBL and share everything we're doing with the community. So thank you for the opportunity. It's very exciting. So as Fred mentioned, our research is sort of evolved in this direction to understand how different environmental variables, especially changes in climate, changes in chemical pollutants in the environment, and how do those different factors contribute to kidney diseases. And the work we're doing is really in the broader context of understanding how environmental change is impacting human health, and really trying to both engage our trainees and students in our program at Duke University, and also trainees on the ground in some of the field settings where we're working in, to really move the needle forward in this type of research that I think is going to become a major area of concern for many communities in the world, not just the ones we are working in. So beyond the work we're doing, it's important for me to highlight that part of this work is really hoping to get some of the new generation of scientists that are coming through our programs to be excited about studying how the environmental change and how the environment generally affects our health. So I'll give a broad overview of our research. I won't go into a lot of detail on many of the aspects, and I'm hoping that anyone in the audience who's particularly keen on some of the details, we can have a conversation. And I apologize in advance if it's too broad at this point. But I want to start with this quote from Time magazine that recently said that chronic kidney disease is poised to become the black lung of climate change. I think this is a bold statement, as Fred said in the beginning when we just joined the call. But I do think it really does highlight why we need to be caring about how the environment is changing around us and how that's going to have health impacts. And at the end of the talk or discussion, we can have a quick vote if you would like on if this is really going to be the case. But I think this sort of topic is captured in the United Nations and World Health Organization's reports recently, really showing that environmental change is directly linked to many poor health outcomes around the world. Particularly in low and middle income communities. But when we think about low and middle income communities, we often think about the global South. But really, we have such disparities within the US, within our own communities, in our own backyard. So really, these health outcomes that are linked to the environment is happening in our own neighborhoods in many ways. And some of the major environmental variables that are highlighted by the WHO include two key components of this. One is to think about chemical pollution, and the other is climate change. And when you look at the numbers of people impacted, just deaths alone, we're seeing about 5 million deaths globally right now. This is between data between 2019 to 2022 that's suggesting that climate change, especially heat stress and cold stress or cold exposure, so extreme temperature events, are contributing to 5 million deaths globally. And then somewhere between 2 to 9 million deaths are attributed to just pollution alone. And that was just 2019. That's a Lancet report that proposed this. And when we think about chemical pollution, we often, I think, forget the fact that there are about 80,000 to 100,000 different chemicals that are currently in circulation that are registered. And you can imagine how many number of compounds that there might be that are not registered, and then how all the different derivatives of these compounds might also be in circulation, especially once those chemical compounds are metabolized. And I think, unfortunately, I hope you're all aware of the PFAS contamination that Maine farmers are experiencing. It's very real and acute here in North Carolina, where one of the companies that produce that compound dumped a whole bunch of that into the CAFÉ River, which is very close to where we live. So chemical composition changes are taking place in our environment in a way that's unprecedented in basically human history. So what we are thinking about generally is how one chemical, and the way the academic structures, the research structures have built, is to really think about how a chemical impacts a certain health outcome, how chemical X goes through a certain biochemical process and impacts your physiology. But what we're beginning to see is that we are exposed to a mixture of chemicals. And then climate change, especially, is acting as a health risk multiplier from those exposure to those chemicals. And that multiplying effect can happen in multiple places. For example, so you may see that with increasing warming seasons, there's going to be increasing wildfires, and I think we're all familiar with this through the news. And that contributes directly to increased air pollution, which is linked to many different number of health outcomes. And then you have parts of the world where increasing drought seasons concentrates both geogenic chemicals, naturally occurring chemicals, but also anthropogenic chemicals in the drinking water sources, as well as water sources used for agriculture. And that increases the chemical burden for the communities that are associated with those water resources. You also see that climate change, especially temperature change, changes the way that chemicals are transported through our environment. So where do chemicals go, where they end up, how well are they distributed and transported across different mediums? All of that is beginning to change with how temperatures are changing around the world. And then that's all at the environmental level. And then we also see interactive effects at the physiological level. So an individual who's working under hot, dry condition might be exposed to a chemical through, let's say, agricultural practices, and the way their body responds to that chemical might be very different from someone who's drinking the same dose of contaminant in the drinking water while sitting just in their house, right? So there are these physiological interactions that we are just beginning to acknowledge. We're yet to understand, but just beginning to acknowledge that might exist. So this way, we think that climate change, especially, is multiplying health risks for many communities. And that is compounded by regions of the world where communities heavily rely on their natural environment for farming and for fishing. And those seasons have been impacted, the yields have been impacted, and that obviously leads to a lack of economic progress and access to a lot of different facilities like healthcare. And that also further compounds this effect of climate change that we are seeing and further exacerbate health risks. And sort of all of that come together has been described as this collision of forces between pollution and climate change, leading to many different health outcomes. And there are communities that are emerging around the world that are termed as climate disease frontline communities. So these are communities that are already showing signs of decreasing and deteriorating health outcomes in their own households and in their neighborhoods, where these two forces are acting in a way that people are exposed to different chemicals differently now, or exposed to heat stress, and that's compounding to manifest into many different kinds of diseases. So an example of that that's really captured a pretty wide scientific audience by this point is this emergence of chronic kidney disease around the world. NPR ran an article on this some years ago saying a mysterious kidney disease slays farm workers in Central America. The Science magazine wrote a report stating that mysterious kidney disease goes global. And then the New England Journal of Medicine indicated that this particular form of kidney disease, which is called chronic kidney disease of unknown etiology, CKDU, so the U stands for the uncertainty around this. CKDU is a quintessential climate disease and a sentinel disease. And that's because this disease captures some of those climate multiplying aspects of climate that's multiplying risk factors. And what we've been doing is really trying to understand how different environmental variables contribute to this disease through multiple collaborations and through community engaged research in several different countries, but particularly in Sri Lanka, which I'll show you in a little bit some of those details. Before going there, I just wanted to quickly show the disease spread around the world. The most established communities with CKDU are in Mesoamerica and in South Asia. So the first diagnosis of CKDU came from El Salvador and Sri Lanka right around the same time, where nephrologists reported that there are people coming into the clinic, they don't have any other symptoms, and they just have chronic kidney failure, very well secured in terms of all the other different types of health outcomes or exposures, health outcomes that they might be at risk of. In other words, they don't really have anything else to worry about, but really they just show declining kidney function and kidney failure. And they didn't know what to do about it. They reported it, and those reports have sort of mounted over the last 20 years, where we are beginning to now see established hotspots in many countries in Mesoamerica, so Mexico, Guatemala, El Salvador, Nicaragua, Costa Rica, Panama, so all of that entire region. And then we're also seeing this in Sri Lanka and India. And now, just in the last 3 to 5 years, we're beginning to see data emerging from countries like Thailand, Cambodia, Vietnam, in several African countries, especially Kenya and Ghana. And then there are suspected cases that are being diagnosed, especially among migrant farm workers, but particularly in farming communities in the US. So there are databases that are being established right now that are beginning to show that people are being diagnosed with kidney failure, and in states like California and Texas, and also here right now in backyard here in North Carolina. Just to give you a little bit of a sense of the disease burden, often 90% of the people who are diagnosed with this, for now at least, seem to be associated with some kind of farming activity. So 60% are farmers directly, and then about 30% are associated with farming communities, and then about 10% of the people would say that they didn't really do any farming work over the last 5 to 10 years. When you look at some of the numbers in places like Nicaragua, they're seeing 1 in 3 men at end-stage renal failure in one community. There's one place we work in India called Uddanam, and there about 60% of the men are impacted and are at end-stage renal failure. So that Science magazine article that said that mysterious kidney disease goes global, they really focused on this community and called this disease the widowmaker disease because primarily men are impacted by it. And then in El Salvador and Nicaragua in the last 20 years, we're seeing about a 5-fold increase. And then about 20,000 people currently at end-stage renal failure that we're beginning to see just in Sri Lanka. So what's unique about this form of kidney disease, different from what you hear about typically in the US, for example, where about 15% of the US community is impacted by kidney diseases. These farmers, so most of the CKDU patients aren't diagnosed with any other risk factor. So they don't, as I mentioned earlier, they don't have diabetes or hypertension. And then often young individuals would die from CKDU. So people at the ages of even 35 have died even within the course of our own fieldwork. So it's really impacting younger individuals compared to the typical CKD that we see in the US. So for us, we extensively focused our work in Sri Lanka and also for several other communities over the last 4 to 5 years. And I would say a little over 5 years now. And Sri Lanka was a good place to start this work because I grew up there, so there was a personal connection. I also speak the language, so it was easy to set up community health work. But also they have a really well-established public health system. So you can actually get records from the last 20, 30 years in terms of health outcomes in local hospitals to figure out when did this disease start showing up in their local hospital. So it's a really good way to look at historical disease emergence and really see that this disease really started showing up only in the late 90s, mid to late 90s, early 2000s. And the other aspect is that Sri Lanka generally has a high literacy rate. So about 95% of the people have had at least a secondary education. So you actually can do public health and community outreach work and increase awareness. And that's really helped in presenting solutions to communities and trying to protect these communities. So for our work, we partnered with a local university there, University of Runa, and also set up a nonprofit on the ground so we can actually do some of the more on-the-ground work with communities that universities aren't able to do always, as well as interface with policy organizations, including the government. Whenever we find something, we can speak directly with the government agents without really having to go through the university channels, which can often become cumbersome and also potentially problematic for the faculty who are involved. And what we've done is really try and set up this sort of broad scale, highly collaborative research effort to really try and understand various aspects of the disease based on defining the exposure, what's called the exposome, all the different environmental variables that an individual is exposed to in the environment. Try and conduct laboratory studies through multiple approaches. My lab particularly focuses on the zebrafish model that I'll talk about in a second, and really got a boost for that work when Ian Drummond was interested in this work and helped us develop some of the techniques, ideas, and continue to help with that. And then we can take that model animal work that we do to the field and conduct studies, population health studies. That really helps us to think about where does the disease start? What are some of the potential biomarkers? How does this disease manifest through life in individuals? So we're trying to do this combination of population health work, animal model work, as well as exposure work to try and understand what are some of the environmental variables these communities are exposed to. And obviously, along the way, we try our best to engage the communities and keep them up to date on what we find, try and build local research capacity, as well as conduct training, both for local university students, but also our students here learn a ton from this kind of work. So what I want to do is to show you a little bit of our data from the last few years. I won't go into all the details as I mentioned earlier, just in the interest of time. So is it okay if I have about 15 more minutes? I hope that's okay. And what we've done is to really try and understand the role of heat stress in this. And as I mentioned, the disease emerges around the tropical belt of the world. And within Sri Lanka, even, you could see that the disease is really localized to the hottest parts of the country. I should say that just because the community is hot, we also don't see the disease. It's the hottest farming communities in the country where we see this. And we all know that through established literature, both heat stress and dehydration contribute to kidney disease, certainly to kidney injury, and repeated kidney injury may contribute to chronic diseases later on, although the data on that is spotty at this point. But it's fairly well established that heat stress can affect your kidney health in several different ways. And we published a review on this a couple of years ago. And the major findings from our work in these communities so far is that if you are an individual who's working extremely hard, engaged in strenuous labor activities in these communities, you're much more likely to come out of it with acute kidney injury. And in some ways, that's not surprising. What was surprising to us is that how little we actually know about how to define heat stress. And I went into this work thinking we should already have our thresholds and everything defined by previous work, but we really quickly realized that we don't really know what the environmental temperature that is stressful to an individual. We know when somebody collapses that that's bad for you, but we don't really have a good sense of defining heat stress, and we especially don't know that for children. And we're just beginning to tackle that. And just to give you an example of this knowledge gap, this is showing temperature data for Sri Lanka, a small island in the Indian Ocean, and how the average monthly maximum temperatures differ across the geographical space across the country. And you can see in light colors, there are some cooler regions, and in hot colors, they're in red colors or darker colors, some warmer regions. And the disease is highly localized to this region. So just because it's warm doesn't mean that the disease is there, as I mentioned. But what's problematic in some ways is that we look at the average monthly maximum temperature, and the thermal profile of that, what that shows is very different from when we look at temperature variance. So how many fluctuations do these communities see? What is the rate of increase has been over the last 10, 15 years to 40 years in these communities? And what are the number of days exceeding the current thresholds, WHO thresholds for outdoor working? And all of those would tell you a different story in terms of what is heat stress for these communities. And even the next layer of gaps that we are encountering now is just because the environment is hot, obviously doesn't mean every individual is experiencing that temperature, right? So we are really trying to figure out how do we correlate environmental temperature derived from satellite data, and how do we corroborate that with on-the-ground monitoring data, but also how do we corroborate with that with individuals' physiological response to that. And this is some of the figures showing that. What you see here in this figure is an instrument that we wrap around our farmers as they work in the field. This work is happening right now in the ground in Sri Lanka, where we are collecting data right at the hottest time of the year during the peak farming season, where we are able to collect multiple different physiological data sets as well as urine samples. So hopefully we can identify what are some of the key thresholds of heat stress for these community members. I won't go into any more details on that, but just want to quickly highlight the two postdocs who are working on this on the ground, and hope I'll get a chance to share this work with you some other day in the future, the rest of that. The other side of this story is to think about chemical exposures. That's how I started my presentation. So there's heat stress and dehydration, but as I mentioned, just because communities are experiencing heat stress, we don't see the disease. You also have to have communities engaged in farming activities. And that's where we also begin to see another burden on kidney health, where these communities are exposed to multiple different contaminants that could potentially pose a threat to kidney health, especially as you're engaged in agricultural activities. So especially in the drinking water, we measure multiple different contaminants. There's fluoride in the water that's at high levels, could be a nephrotoxic compound. There are heavy metals. All of them are at lower levels, but it's there. We measure arsenic, cadmium, lead. We also find that the water in these regions are hard water, so there's high hardness in terms of higher levels of calcium and magnesium in the water. And we also measure several different pesticides in the drinking water. And just to get a better sense of this chemical exposure in these communities, we've now taken two different approaches. One is to really identify and characterize drinking water sources for community members who are impacted by the disease. And the other part that we've just completed is to put these silicone wristbands around our farmers before and after farming season, and also during their farming season, where these wristbands can capture essentially volatilized chemical compounds, especially organics, in their wristband, and we can analyze them in the lab. So all of this work is done in collaboration with Dr. Heather Stapleton, and also Professor Lee Ferguson, Professor Stapleton, Professor Ferguson, as well as students in our lab and Lee's lab, Sakina Shahid and Jay Kulrick. I won't show all the data. What I will show is that the drinking water measurements is allowing us to think about a potential source of exposure for people, and also a snapshot of what's in the environment at that given time. And we recently published this work that examined about 200 different water samples, comparing communities impacted and unimpacted. And what we ended up concluding was that there are several different compounds in the drinking water samples of the people impacted by the disease. And those are vanadium and high hardness in the drinking water, fluoride, and also glyphosate, which is something that I think many folks in the US and around the world use, because it is actually the most sold herbicide in the world. And it's heavily and extensively used in these communities. And what we find is that the way that glyphosate should be removed from the environment isn't actually what's happening in these habitats or in these drinking water sources, because of the high hardness. So glyphosate is being stabilized in the environment because of the hard water. And in addition to that, we looked at a number of different other chemical compounds, and essentially what we find is that there are multiple different sources of nephrotoxic chemicals in the drinking water. But the hard part, and this is a major challenge in environmental toxicology right now and environmental health right now, is when you're exposed to chemical mixtures that are these complex mixtures that have these interactions, like the way that glyphosate interacts with heavy metals and other cations, it becomes incredibly difficult to disentangle what chemical is most nephrotoxic. And this is really where the model animals have become helpful to us. And we've been able to get some funding through the National Institutes of Environmental Health to really develop a study that compares how multiple different model systems respond to some of the suspected nephrotoxic compounds, and how does the kidney function change with exposure to those compounds. And the work that we're doing in collaboration with Ian is really focused on zebrafish, and Ian's part of this consortium. And there are several other folks who are involved in comparing the data we generate in the zebrafish model with mice and with 3D cell cultures or organoids, and also with 2D cell cultures. And then the hope is that we can compare our chemical composition data with our phenotypic data to try and isolate what type of compounds are most problematic. We've done some work on it, and I won't really show you that data here. But I just wanted to quickly highlight that Ilaria Merodka, who's a grad student in the lab, is really leading this work. And she's using the zebrafish model in sort of three different ways. One is to think about the zebrafish model as a screening tool. So we can bring in these water samples from the field, conduct high throughput exposure studies, and really try and understand what types of chemical signatures are always associated with nephrotoxicity as well as other toxic outcomes. She can then use that information. She is using that information to do long-term exposure studies to really understand how both kidney function and also the long-term kidney health are impacted from exposure to these suspected chemicals. And she's really focusing on glyphosate here, as well as the metal mixtures that we find in the drinking water samples. She's also, along the way, found that these metal mixtures and glyphosate can be quite developmentally toxic to the kidney. She's found some really neat data that indicated to us that these drinking water samples could, in fact, affect kidney development in the zebrafish model, which really made us want to go and re-examine what we know about the disease in a different way in the communities. So just to take you back to the community, we learned from the zebrafish model that perhaps there's a risk to children from the drinking water samples in these regions that we've collected these samples from. And I'm quickly skipping over one slide to show that we established a 3,000 children cohort in these regions, in these impacted, unimpacted communities. And what we find is that children who live in CKDU impacted villages already show signs of poor kidney function. This is based on several different markers, but especially this one marker called kidney injury molecule 1, or KIM1. And we are following these up both to monitor their exposure to environmental contaminants, but also to monitor their response to heat stress. Because what we find is that there are communities where there is the same chemical signature, but there is no CKDU. So we do think that there's an interaction going on. So we have our kids, the children who are part of the study, also wearing the heat stress monitors around their chest as they go to sleep. And then also they play, and they especially play the most wonderful sport, cricket, in these regions, where they spend several hours outdoor in the sun, although they don't do a whole lot of moving around in that time. But we can get the sense of the levels of heat exposure that these kids are experiencing in those communities. So this is ongoing parallel work, especially through our team on the ground in Sri Lanka. So I'm going to quickly wrap up around here and say that we are barely scratching the surface of CKDU in many ways, but we've been able to identify that there's several different risk factors for these communities that might increase their susceptibility to other risk factors as they grow older. So especially drinking water poses a threat to kidney development. We've confirmed that with our model systems, and we're seeing that with our children, and long-term studies will help us better identify how persistent that is. And also then will help us think about how risk factors like heat stress further exacerbate the impact on the kidneys. And broadly, this work is really helping us to think about how we study chemical mixture toxicity, how do we study interactions with heat stress, but also how do we define heat stress, and think about some of the data gaps in that space, and hopefully try and understand mechanisms of early kidney damage, kidney injury, and then hopefully we can integrate some of the model animal work with population health work to better inform questions around CKDU, but also I think in many ways CKDU might just be the tip of the iceberg when we think about how environmental changes is impacting kidney health. So this is a report that just came out, and the president of the International Society for Nephrology said that kidney disease stands as the third fastest growing cause of death worldwide, with deaths attributed to kidney diseases surged by 50% from 2000 to 2019. So there's a clear rise in kidney disease incidences. There's a clear rise in number of admissions related to kidney disease, especially during heat wave events. So all of that is pointing to us that perhaps these communities that are impacted are really at that most unfortunate stage where they're really showing the chronic disease stage, but maybe we are seeing we're not able to see it yet in these communities, but maybe that's something we should be thinking about. But the biggest challenge in front of us, especially as environmental health scientists, is to try and understand long-term health outcomes, including kidney health outcomes, following these lifelong exposures to multiple environmental stresses. And I think again, I'd like to highlight that this is not something that's unique to the global South. It's something that's happening in the global North, in our own communities. And I think we can do a lot more just from the little we know already to prevent manifestation of these types of diseases in our own community. So with that, I'll thank my lab group and the group that's really been helpful on the ground in Sri Lanka, as well as some awesome collaborators that I've been fortunate to work with. And happy to take any questions. And I'll leave with this last slide where sort of everything, all of this work, the inspiration, all of that started at MDIBL many, many years ago, 2 decades ago now, when we had our first molecular biology class, short course as College of the Atlantic students with David Toll, who went on to become my mentor for many years after that. So thank you.
Yay.
That was a pretty extraordinary example of combination of so many different disciplines under the rubric of community health, but right down to population health to molecular pathways, and so it's kind of stunning. And I just want to mention to everyone that if you have questions, you can write them in the chat box on your Zoom, or you can raise your hand physically, and we'll look for you, or you can use the little raise your hand marker on the Zoom application. Jane Disney, Dr. Jane Disney, who directs the Community and Environmental Health Laboratory here at MDI Biolab, has already chimed in with a question, Nishad, and I'm not sure if you can see it. So, but for everyone, I'm just going to read it. And Jane, are you with us? Can we just open the mic for your?
She just took her camera off.
Sure, yeah. Hi, Nishad.
Hi, Jane.
It's so exciting to hear from you and to see how much progress you're making on this front. I'm just venturing into, because how can you avoid venturing into some work with PFAS? And I'm wondering, is anyone able to pick up PFAS exposures in these silicone wristbands, or how, is there any other way to do it besides just doing, a whole blood serum? Or, I mean, everything else seems so invasive for people.
Good to see you as always. And thanks for that question. As far as I know, I don't want to speak for Heather, of course, but I know she's trying to figure out how to pick up PFAS in the wristbands. I was part of a pilot run where I was wearing a wristband for a bit, and I know they were trying to measure some of the PFAS compounds, but I don't think it's been a straightforward road so far. But hopefully road ahead is less challenging. I do think part of the problem is that the way that the wristbands work, the compounds have to be volatilized, and it's sort of aerosolized exposure they're getting. So unless you are washing your hands and really dipping your hands in a PFAS containing water sample, let's say, I'm not sure if you would pick up just from air. I think in many ways to define the PFAS exposure, I think passive samplers might be the way to go, as opposed to wristbands that are specifically targeted to capture PFASs.
What kind of passive samplers do you mean?
So if you're trying to think about water, so I was more thinking about water sampling. So you could do passive water collections and then try to do the same type of LCMS analysis that you would do. And
Interesting. Thank you.
Thank you,
sorry,
Jane.
there's no good answer to measuring PFAS. It's been I guess, the bane of everybody's existence, right?
Well, but it is a fascinating way to try to track what's going on, just these wristbands. And I'm curious, how big are the populations that you're working with in Sri Lanka?
Depends. So some of the data that I showed come from a cohort of 200 people to the children work is 3,000 children. So the adult work is often limited to smaller cohorts because you can clearly do these case control studies, who have the disease, who has the disease and who's not diagnosed yet. Whereas the kids, we're just beginning to understand how to even measure kidney health in a child who doesn't have genetically predisposed kidney disease outcomes, or straight CKD, chronic kidney disease already. So identifying early kidney injury has been challenging. We just held a symposium here at Duke in our program to really start getting people together who are pediatric nephrologists and environmental health scientists, and some of the work we're doing to try and understand what are some of the markers we can use for monitoring children's kidney health. But because of that, we need large cohort sizes.
What's it like getting buy-in from the communities that you're working with? If some of your work touches on touchy issues like pesticides for these people's livelihoods, is that a difficult prospect, or do you find that you've got the techniques that are needed and able to get them to participate and feel good about it?
It's a great question, and I think perhaps 2 or 3 questions embedded in there. One is thinking about what does it mean to study with a pesticide like glyphosate, which is something that everybody wants to use and has used for a long time. And there's, as you can imagine, a lot of pushback. When we published the paper, we had 2 rebuttals we had to respond to. I don't think I've ever had to write a rebuttal in anything else I've ever written. And there was a lot of press, and especially in Sri Lankan press, about different scientists arguing over the role of glyphosate. So it left us somewhat in a challenging position, because we didn't want to claim at all that we need to stop using this pesticide. Our claim was, in fact, that it's persistent in the environment in a different way than what initially was thought, and that we just need to we were really focusing on the idea of we need to provide clean water to the communities as opposed to banning these chemicals, right? So there are different, so there's a different challenge there in thinking about community buy-in, because we were trying to work with the community who needs the glyphosate so they can do the farming, and in their words, they would say, I need to use my pesticide to live until I get my kidney disease, right? So they would not want to not use this chemical compound. So we were trying to be very careful in there. But otherwise, overall, we've had really good fortune to be part of these communities and to work with them year after year. And we have field teams who go there quite often. The schools have been extremely helpful in getting participation. I was inspired by Jane's work that looks at that takes school students into her work in terms of bringing water samples. We were doing some very similar work there, where the kids would bring water samples from their household, and we'd measure their kidney health. And we've never had any pushback. The biggest issue there is no one wants to give a blood sample. So we've always had to figure out ways to come up with better kidney health markers through urine, which is often not the best. But I also don't want to push for more invasive sampling procedures. So that's been a challenge for sure. But otherwise, it's been a great experience, and feel very lucky and fortunate to be part of those community efforts.
Where should we take what you're learning about this specific community and how climate change and drought and pesticide and heat stress all create this new kind of complex health equation that we need to pay attention to? How do we apply it to the US or to any other place around the world? Are you getting to the point where you can sort of think about screens and say, we're seeing this kind of combination in the the broccoli fields of the South, or I don't know what, but is that something that's starting to go on? Is it something you think about? How do we apply this to people's lives and communities elsewhere, including here in the US?
Great question. So I think twofold. One is that already there are communities seeing increases in kidney disease outcomes in, for example, in Texas, where Dr. Sheikh Hamad, who's at Baylor College of Medicine, he had to basically find his way to find funding to set up a dialysis clinic for migrant farm workers who were coming in with kidney failure to the clinic, and they had developed it since arriving in the US. And then there are farm workers in California where there's a cohort that's being set up currently. And the lead PI there, Shuchi Anand, who's also a nephrologist at Stanford, and she would say that kidney health is a barometer for climate and pesticide or climate and pollution-driven health outcomes. And instead of monitoring for the environment, we should be monitoring for kidney health in these communities. So when you say, what should we be screening for? Perhaps we should be paying more attention to just general kidney health outcomes in our community, especially communities that we know are vulnerable to heat stress, vulnerable to environmental contaminants. Obviously, it's easy to say. Insurance will have a lot of say in how that's done. But that's definitely, that's also what this new report that ISN, International Society of Nephrologists, put out, saying that we will not necessarily be able to understand all the different environmental factors contributing to this disease, but we can definitely screen better and come up with better awareness systems to improve kidney health outcomes in the US and globally.
We have a question from the watch party at Patricia Hand's house. So if you guys are able to open up your mic, we'd love to hear from you.
Yes, it was fascinating, and I'm very impressed with the research, especially looking at the whole community focus. But are there other chemical compounds like glyphosate that may have a similar makeup that should be kind of carefully watched? That it may have similar chemical aspects?
That's a great question, and thank you. Off the top of my head, I don't know of any other chelating organophosphate. There are definitely other compounds that are known chelators of metals. I don't think I know of a pesticide or specifically an organophosphate that can act as a cation chelator, where basically glyphosate itself becomes a stable compound, and then also acts as a carrier for these other heavy metals like cadmium or lead or vanadium. And we do think that that poses a greater threat to the kidney, because that seems to be where once reabsorption starts to happen in the renal tubules, that might be where the metal burden most increased in terms of a site of exposure within the body. We don't really know why. We just have zebrafish data that shows if you look at fish that are exposed to glyphosate and metals, kidney tissues are the most targeted tissue during development. And if you just have glyphosate, actually targets neurodevelopment, not kidneys at all. So there's sort of 2-3 questions to think about. One is, how sure are we that chelation is what's driving this persistent toxicity effect? And then what other compounds can actually do that type of chelation?
That's very helpful.
Dr. Jayasundara, I want to encourage people to raise your hands and ask your questions, because soon we'll run out of time in just a few minutes. A little bit of detail question, and then I'm going to ask you maybe to talk about some other things that you might be working on. But just in there, you mentioned that there was a profile of chemicals which in one group of children you did associate with early signs of kidney disease, and then in another that you did not find that. And you're looking at heat stress as the compounding factor that makes a difference. Did I understand you correctly?
Yes.
And what is it that gives you confidence that heat stress is the right suspect to be pursuing at this point? When I understand that there must be certain factors that you've already figured out that say, well, it's a likely one. What's the world of possible factors that may be competing with heat stress for making that difference between one of those, that one healthier population than the other?
Yeah, great question. And I think that that's true for both children and adult communities in terms of we have the disease endemicity really unique to tropical farming communities in the world, as I mentioned, right? So the third factor that's been discussed quite a bit is that if you're in the tropics, yes, there's heat stress. If you're farming, you're exposed to chemicals. Is it changing the potential exposure to other infectious vectors? Or, for example, leptospirosis was discussed as a potential contributor to kidney disease, or CKDU specifically. And then also, is it changing the cyanobacteria and cyanotoxins in the drinking water? And that's also been explored quite a bit. And those efforts haven't really come out with any conclusive evidence to show that there's a role for those. The two factors that continue to stand out is that if you're living in a hot farming area is where you see the disease. So when we set up our children cohort, we made a conscious effort to make sure that we have children representing multiple types of communities. So communities that are most certainly farming through the country, but also communities that aren't farming, but just hot. So those are the countries in the coastal regions where there's extreme temperatures that seem to occur much more consistently in those regions compared to the middle of the country. And then children who live in farming areas that are not hot, right? So sort of three different variables. So hot and farming, hot and not farming, and then farming and not hot. And just the preliminary work, this is a grant that's currently in review, hopefully we'll get funded, is indicating to us that those two factors will have to come together to actually see the disease popping up. But how it is doing that, we have no idea. And to your question, we do know heat stress does cause kidney injury, but is it what's really driving this disease here? We don't know. In other words, could it be that heat stress changing the composition of a certain chemical or leading to population of bacteria to grow faster that's actually producing some nephrotoxin? That remains a possibility, right? That's where we really need to better understand what heat stress does to us in terms of kidney health outcomes.
Okay. Fred, I think we have another question from the watch party.
I don't want to take too long here. I was just curious whether there were any neurological hotspots also associated with this. Sometimes there've been with cyanobacteria and other related compounds, you find neurological damage. I was just curious about that and the heat combined with neurological impacts.
Yeah, that's a great question and not something we've looked into in detail. So at this point, we don't know. There's a lot of interest in it. I know that the Duke University especially is very interested in understanding neurological outcomes as well as mental health aspects of living in these communities or the lived experience, but also exposure to these contaminants and potentially the same that causes kidney injury. Could they be affecting neurodevelopment? I think hopefully there are going to be some answers to that in the next 5 to 10 years, but I don't see it happening any sooner than that. But I think it's an important area to look at for sure.
Nishad, oh, sorry, Fred, you're muted. In case you wanted to unmute.
Oh, sorry about that. Thank you, Jerry. I just wanted to mention that folks were at Dr. Hand's house, and when Dr. Jayasundara mentioned having gone to his first science sort of or laboratory-oriented class when he was at the College of the Atlantic at MDI Biolab, it's to some great degree thanks to Dr. Hand's work bringing up the INBRI network, the IDEA network of biomedical research excellence, which is something that the lab is getting ready to go into a next phase, hopefully, with the blessings of the NIH, and we're waiting for the word on that. But it's been very exciting for me as a new person at the lab to observe this educational opportunity. So Dr. Ham just won a nice award recognizing that work. I'm curious, was that your only INBRI experience, Nishad, or Nishad, did you have more education or more support from that program as you developed into this wonderful scientist that you are now?
Yeah, thank you, Dr. Han. And yes, I think I used every opportunity to use INBRI and BRIN Maine funding. So I think I got every one of those available funds along the way. So I think first it was BRIN Maine which funded the course, and then it became INBRI. So I was an INBRI summer intern. And then I think when I came back to Maine as a faculty, I was able to get funding through INBRI to both support research, but also support students in my lab. And so I think I've used that in every way possible.
Well, it was money well spent, Nishad. You're doing some amazing work. And I think for those of us who have been around a while, it's so much fun to see, that full circle story. I think when you were at the lab one year, you brought someone, as you mentioned, that had a student in your lab who had INBRI funding, which I find just so poignant that you were able to kind of complete that circle. So thank you for this amazing work. It's really, really exciting to hear all that you're doing. And I'm just really curious. You mentioned, some colleagues on the West Coast and being aware that kidney disease is kind of a sentinel for climate change. Are you aware of any studies like the one you're doing that are happening in the United States, or similar attempts to try and understand the impact, particularly on farm workers?
Yeah, I think these efforts are just starting, as in some are starting this week even.
Oh, wow.
So the symposium we just had last Friday was a gathering of scientists who are just beginning to look at how environmental exposures, particularly heat stress and maternal exposure to different chemical contaminants, contribute to pediatric kidney injury and also adult kidney diseases. So I think there's growing interest in this, and there's going to be a North Carolina cohort that's funded through a pilot funding source from Duke, and there's the cohort that's in California, and there's one in Texas that's actually funded by an NIH grant right now. And then pediatric work is just beginning. So there's a nephrologist, pediatric nephrologist, Kier Sanderson, who's at UNC, who's doing some amazing work to try and monitor kidney health outcomes in children where their mothers were exposed to different metal mixtures. And they're already seeing that that's directly correlated with declining kidney health in those kids. So this is just beginning work. And I think the work from these communities are in many ways a sentinel in that sense, in pointing to something we should be paying attention to here.
Can I ask one follow-up question?
Oh, sorry. A comment or question.
Okay. Yeah, go ahead.
Chad, it was an absolute tremendous pleasure to have you in that first cohort of students. And David Toll was the first faculty member at MDIBL who started those short courses and who mentored you over many years, I believe.
Yes.
Betty, his wife, is with us here today. Hello, Nishad.
Oh, hi, Betty. Sorry I didn't see you earlier.
To hear about your research and, to see how the BRIN and INBRI really made an impact on you and on us. So thank you.
Thank you. No, thank you so much.
Jerry, I think you had another question, and anybody else, now's the time to get it in because we're pushing up on an hour.
We are pushing. So I just quickly, I'm curious, Nishad, how you're finding funding for this. You mentioned National Institutes of Environmental Health Sciences. I mean, has it been pretty well received? Are you finding that, grants are never easy, so I won't say that, but I just wonder how difficult it's been to get this project, fully funded.
Yeah. No, it has been extremely difficult. So a lot of this work was thanks to funding from World Bank, and then followed by a lot of pilot funding from Duke and the Duke Global Health Institute and the Nicholas School. They've actually been incredibly generous in making sure that this cohort studies continue. NIH is much more keen on funding work that has direct, tangible molecular level results or tangible data that could lead to hopefully some kind of a cure or disease mitigation method, right? So it's been challenging to get funding to do the population health work. We did get that Victor funding, which obviously had to be presented in a slightly different way and really highlight that low-level environmental contaminants during exposure, not exposure to low-level environmental contaminants during maternal windows of child development is critical for kidney health, right? It's really showing that correlation helped us to get funding. So I think as the area, this area of research is growing and people are becoming more interested, hopefully there'll be more opportunities. But my hope is that we can keep pushing the zebrafish work forward because we are, I think, starting to recognize some pathways that we could be following up as mechanisms of disease initiation and progression. And I see Ian's thumbs up, and I just want to say, I can't thank Ian enough for really giving us a lot of resources to get that work started and continuing to be supportive and even just offering advice and help at all times. So hopefully that work will lead to better funding. I do think that the global health workers, we're trying Gates and Rockefeller and other foundations, but my hope is that there'll be other entities that would be able to support that work.
Well, we wish you all the best. And, just a plug for MDIBL. What we say is that once you've been connected to MDIBL, you're never allowed to leave. So we will continue to follow and hopefully, watch your collaboration grow. So it's so much fun to hear this update. Thank you for sharing it with us.
Thank you. Always an honor and a pleasure to be part of all of this effort. So thank you.
And I also want to thank Dr. Jayasundara for being so helpful in putting this together. I just want to mention before everybody signs out that's in a month on May 13th, the next Science Cafe has a title called Coastlines, Sea Level Rise and Collaboration on Mount Desert Island. It's a continuation of a project that's been spearheaded by the MDI Historical Society and the Skudik Institute to sort of take a cultural and artistic look at the meaning of sea level rise and observations. And they've brought in new partners such as the Gulf of Maine Research Institute and the Maine Coast Heritage Trust now, and it's going through some new iterations. And that group is going to be on our next Science Cafe to talk about where that's headed. It'll be with the MDI Historical Society Executive Director, Rainey Bench, and the Skudik Institute's Science Communications Specialist, Catherine Schmidt. And so we hope you'll be able to join. For people who might be watching a recorded version of this, well, we have many friends here today. You can find out more and keep up to date with us by signing up for our electronic newsletter, which is at mdibl.org/support/ learn. Oh, I can't remember. It's
learn more.
Stay informed, stay-informed. So I'll say it again, mdibl.org/support/stay-informed. So thanks everyone for joining and look forward to joining you all at the next one.